thank you all for being perfectly in time. That's wonderful and not so easy on a Monday morning, 8 o'clock, but I hope you will not regret it and learn something today about transcription. And maybe you wonder why I have this photo up here of our beautiful Paulinerkirche in Göttingen. I don't know if anybody has been at this event about 1 week ago, 10 days ago, where we had Ugo Shahin and Özlem Türeci of BioNTech with us. And we actually discussed this special year 2020 when they made the first corona vaccine within 10 months. It was an incredible evening. So interesting to hear how it became possible and all the science behind it. And the reason why I show it here is to remind you of the power of messenger RNA, right? So they inject micrograms of messenger RNA into your arm, and it causes this incredible immune response that protects you for several months quite safely from the coronavirus. And why is that? It's because this mRNA has the potential to make a protein, and it can make a large quantity of proteins if it's an mRNA that is optimized for protein production. And the process of transcription, of course, is the fundamental process that makes all these messenger RNAs in our cells, right? And today we want to learn how that is possible, how transcription works. Also tomorrow we will dig more into the details. And I want to start by giving you an overview of what we are going to discuss over those 2 days. Today we start with general aspects of transcription. First half hour or so we spend on structural biology methods. Why is that important? Because when we have structural information, we understand the mechanism of transcription. And then I talk about the structure of the RNA polymerase, the central enzyme that uses DNA to produce messenger RNA. And then later this morning, about the initiation of transcription and how it can be regulated. And then tomorrow we talk about functional genomics, so fundamental methods to investigate transcription and its regulation in cells, right? And then we talk about the elongation phase, how the mRNA chain is extended, and also a little bit more about the regulation of transcription. So just as a reminder to you how important transcription is for life, this is a mouse embryo, and you see the staining of several different transcription factors here during embryogenesis. So transcription factors determine which tissues you form, at which time and where you form them, right? This is because transcription underlies the development of organisms, the differentiation of cells, and of course it's also important later in life. For example, responses to the environment as the immune response that I was just briefly mentioning, they are all regulated by transcription. So a really fundamental process. So it's worth to be able to understand how it works. Okay, let's get started. Maybe before we get into things, just a brief introduction. My name is Patrick Cramer. I've been here for 9 years. Before that, I've been in Munich for 13 years or so as a professor at the university. And before that, I've been abroad for 7 years, France, England, California. Our interest is indeed transcription. There's a few people here from the laboratory. You probably get to know them later as mentors. You can ask them any question you may have about transcription or life as a PhD student and so forth. We have been working on the structure and function of RNA polymerases over the years. And I think we did learn a fair bit about how transcription works. And some of this I want to share. But we have to start with the basics, right? So let's start with the tools that we use to investigate the transcription machinery, how to visualize the transcription machinery. Why do we do structural biology? Well, all the proteins are made from amino acids, like all these tools are made from steel, right? But all those tools have very different function, and proteins also have incredibly different function. And the reason is They have a different shape, they have a different structure. And it's important to know that structure in order to understand how proteins fulfill their function. First of all, we need to make a protein that we wish to study structurally. For example, using large fermenters like the one we have here at the Institute, where you can grow 100, 200 liters of cell culture, but also overexpression, and then you may need a smaller amount of cells to get enough protein. And then we purify it using chromatographic systems that are actually nowadays controlled by computers, so you can also program experiments you want to run on those machines overnight. Then once you have your protein of interest, the classical method is of course to crystallize it and to do X-ray structural analysis. So you form these little drops of 1, 2 microliters, nowadays actually even less, a fraction of a microliter. And at certain concentrations of the protein, when you have a saturated solution and you go into oversaturation, the protein can either precipitate, or if you do it properly, if you do it slowly, it can form beautiful crystals. And then those crystals can be used for X-ray analysis. So what is that? It's actually relatively simple. The concept is relatively simple, but when you dig into the mathematical details, it becomes complicated. But today I just want to convey the principle so that everybody has a rough idea. And the principle is that you take the crystal, you flash freeze it to preserve it in liquid nitrogen, and then you shoot it with X-rays. You need extremely strong X-ray beams like the ones that are produced at synchrotrons, very large machines that you have at different places in Europe and around the world. And then the crystal, because it's actually made of a lattice of protein molecules, will diffract those X-ray beams in different directions, right? And you can use a detector to record those diffracted X-ray beams. They will leave a pattern on the detector. And from that, you can actually calculate what is called an electron density map, which is a 3-dimensional map of where the electrons are. And once you know where the electrons are, you can build an atomic model that explains these densities, that explains this electron cloud. And this model you can then refine, and you can actually get to an atomic model of your protein. Now, just briefly how it works nowadays, a lot of it is automated. You have, for example, robots for the crystallization. We used to pipette all these droplets by hand, but nowadays you can do 1,000 or multiple thousands of experiments using robots. And then you have so-called hotels where the crystal plates with the little hanging drops are stored, and you can visualize these droplets under microscope in the hotel. You can do that remotely from home or wherever. You can log in and see whether your crystals grow over the next days or weeks. You can also then shoot those crystals at home with a so-called PX scanner. It produces X-rays, and it lets you decide, is that really a protein crystal, or unfortunately only a salt crystal that formed in the buffer. But once you have protein crystals, you can send them to synchrotrons, like the one here at ESRF in Grenoble, the European Synchrotron Radiation Facility. This is actually the place where I did my PhD, very beautiful, surrounded by the French Alps. And when I did my PhD there, it was a brand new machine. It was very exciting, to see how the beamlines are set up and improved. And at that time, of course, you would put your crystal in by hand and, do everything by hand. But nowadays, you can ship your crystal by FedEx in a frozen state, in a dewar, and it will be put on by a robot and basically measured from remote. And you can also then get the data back. You don't even need to travel there. So if you're lucky, if you have a good crystal, you get a beautiful diffraction pattern. And each dot here is actually one diffracted X-ray beam. It's caused by one diffracted X-ray beam. And so the information you get here, you can easily see you have dots that have different intensity. So there's a lot of information in the intensities. But also, of course, each diffracted X-ray beam is found at a certain position, right? And from that information, you can use that information to calculate electron density maps. What you also need are the so-called phases of the diffracted X-ray beams. And that is more difficult, but this is, you've probably heard about that. This is why we use either selenomethionine labeling, or we use heavy metals to derivatize the crystal. Then we can change those intensities and we can draw conclusions about the phase information. And once you have estimated phases, you can calculate those electron density maps. And that is a great moment when you actually see the electron density map on your screen, because you know you have solved your structure and you can now build an atomic model into these clouds. And much of that is also now being automated. You know, it used to be that you wake up in the night and you realize, I tried for the whole day to fit in this protein chain, but I actually did it the wrong way, and now I know, and you go back to the lab and you can build the protein and it fits in. So it used to be a challenge that could be solved only by the human brain, but you know, programs are getting better and we have more information about proteins. But just a little, tiny little bit of history, because it's so important for molecular biology as a whole, you know, how structural biology evolved. Here you see John Kendrew, who solved the first protein structure, actually together with Max Perutz. John was working on myoglobin and Max was working on hemoglobin, both in Cambridge. And this is the way they built models at the time, you know. They really put the individual atoms on these sticks to make a three-dimensional model. Must have been, you know, very, very difficult at that time, also with, you know, no real computer power as we have it today. And please also remember the DNA structure. And remember Rosalind Franklin, because she produced X-ray diffraction patterns, not of crystals, but of fibers of the DNA. And you know, if you have such fibers of DNA and you shoot X-rays, you also get a pattern. You don't get the beautiful, you know, very complex diffraction pattern. You only get that with crystals. But if you have regular fibers with certain distances, molecular distances in those fibers, you also get some patterns. You've probably seen this cross-like pattern, this X-like pattern that she obtained. And that was really the key, that was the crucial information to find out that DNA is a double helix and that also the two strands are actually running in opposite direction, that they're anti-parallel. So basically her contribution is to provide this key experiment. It was stolen from her, as you know. It's a long story. You can read about it. For her 100th birthday, I actually wrote a little piece, just 2 pages. It's very interesting when you recapitulate the story. And then the story is that Crick, actually, Francis Crick was the one who realized that the two strands have to be in opposite direction because of the symmetry in Rosalind's diffraction pattern. And what James Watson did is basically that he played with the bases and he found the base pairing. And then these 3 pieces of information led to a model. But it's important to remember that the data were from her, and I don't think that until the end of her life she knew that it was stolen in this way. There's other methods, of course, as you know, to solve structures. There's nuclear magnetic resonance, which also gives you dynamic information about proteins. And there's electron microscopy, which over the last 10 years or so has developed really dramatically. You probably all know there was a Nobel Prize mainly on the development also of the direct electron detectors. And that was really game-changing, because now electron microscopy would also routinely allow you to get to this level of detail that before only X-ray crystallography could give you. So now we can routinely also solve atomic level structures using cryo-EM. And there's a little movie here that shows you how cryo-EM works. You have your protein in solution, no need to crystallize it. You put it on this grid and then it's flash frozen, very deep temperature. You prevent the formation of ice. And then the protein is suspended in this very, thin layer of liquid, which is held simply by the surface tension. And you can then, bring this grid into an electron microscope, shoot electrons at it, and you will then see individual images of these protein complexes. And since they will be, different from each other, you have to sort them in the computer. You have to find those particles that resemble each other. And when you then have enough of those particles, you can do a so-called back projection, and you can solve the three-dimensional structure of the protein that you had in solution, right? So this is given here again in a schematic. You have your EM grid. This is only a couple of millimeters, so very small holes in this grid. And then inside of these little holes, you have your protein of interest embedded in a buffer solution, a very thin solution. So this is held just by surface tension and then flash frozen. And now you can image these individual proteins. And you can imagine, when you have proteins in solution, of course, they're moving, right? They're moving around. So now you flash freeze the grid. And so the proteins are then stuck in a certain orientation. But of course, the orientation is not the same. It will be different for all proteins, right? There may be a certain bias because proteins may be, because they are elongated or so, they may tend to be in one orientation. That can cause problems, but often you have different orientations. And that is actually very important because that allows you just by imaging from one direction to see the protein from different sides, right? And once you see the protein from different sides, you can also reconstruct the volume that underlies the formation of these images. And this is seen here, the electron beam comes from the top. The protein complexes are in different orientations. You get these different images, and then you sort those images in the computer, and you define the orientation. And once you have the orientation of the individual particles, you can back project and then get a reconstruction of your original three-dimensional volume. This is the principle of cryo-electron microscopy, I should say of single particle cryo-electron microscopy. Now, how does it look in reality? This is, how it looks in a textbook. But I just gave you here an example from our work so that you see how it looks in reality. So, you make a huge effort, biochemical effort, right, to make the best possible protein sample. All the protein subunits in your complex are stoichiometric. It all looks beautiful. And then you do the experiment. And of course, there's stress, right? You flash freeze. There's also this very thin layer where there's the surface tension actually produces force on the protein. And so then what happens is that often these complexes fall apart, right? And so when you sort the different particles, you can find all these different species here. Some of the particles have lost this density, others have lost the blue density and so forth. And so it's really then a big effort to sort everything in the computer, because you will only get a high resolution structure if all your particle images are images of the complete particle. If some particles lack this subunit and others lack this one, it will be very heterogeneous, and when you do the back projection, you will get very noisy maps. So you have to do a very careful sorting, and then eventually you end up, with structures that are, at a reasonable resolution, 6 angstrom, 5, 4, even 3 angstroms or better. And when these methods were established, our lab was all crystallographic, and then we moved to cryo-electron microscopy. And then what was so interesting to see, when we take the two different samples, in this case, it's RNA polymerase II, this of the same enzyme, but one, for crystallization, the other one for cryo-EM. How does it compare in the end? Can we get to the same result? And this is just a tiny little piece of the polymerase, and you really see how similar the maps are. One is by X-ray crystallography, the other one is by cryo-electron microscopy. So that was incredible, and ever since, we have shifted the laboratory, and now we use mainly cryo-electron microscopy for structural determination. But how does it look like on a global scale? How did this field evolve? So yesterday I just, for fun, went to the protein data bank and I looked, are we already at 200,000 structures? We're actually close, you see, very close to 200,000 structures deposited in the protein data bank. That was founded over 50 years ago. And it's very, very, very important that the community, the worldwide community, cooperates, agrees on where to collect the data and on the format of the data, and then to deposit the data so that everybody can benefit from this. And you will see in a second why this is so important. I'll give you an example. But just to show you that the vast majority of structures really comes from X-ray crystallography. The reason why we know about the hundreds and hundreds of different folds and protein structures is X-ray crystallography. NMR contributed also by solving complicated domain structures that would not crystallize and so forth. And now, of course, cryo-electron microscopy is taking over. And when you look at that maybe in 10 years or so, you will probably see a much larger fraction of cryo-EM structures. Now, why is it so important to collect data? Of course, if somebody's interested in a certain protein, they can just download the structure and it helps them. They can design a mutation and so forth and so forth. But what is also important that once in a while you get a paradigm shift. And this only happens when you have reached a certain level of understanding or when you have accumulated a certain number of data sets. And you've all heard of the AlphaFold software by DeepMind, which is actually now a daughter of Google. And if you look, over the years, this is the structure prediction competition, a worldwide competition of research groups that try to predict three-dimensional structure based on sequence alone, right? And for years and years, basically the community was stuck at some 40–50% of accuracy of the modeling. And then suddenly, 2018, everybody was very excited about something called AlphaFold, which uses machine learning to predict structure. And then, 2 years later, there was a huge breakthrough. AlphaFold 2 basically can predict most protein structures or folded domains, of course, with an accuracy that is at the level of individual amino acid side chains. So it's like a high-resolution crystal structure. This was really, game-changing. And so I'll give you a few examples here. These are some of the first examples when it came out, about 1.5 years or so ago. So you see here that in blue you have the computational prediction by AlphaFold, and in green you have the experimental structure. So it's virtually identical. It's really incredible how it works. And how do we know that this is a real prediction and that there was no information from the experiment? The way it works is that in this competition, there's protein sequences given out to those theoretical groups that are making the predictions, where we know there will be a structure, but it's not published yet. And then, a few months later, when the predictions have arrived, the structure will be revealed and then can be compared in an unbiased manner. And this is how well it works for AlphaFold if you have small folded domains. So again, I've written a little, this is only 1 page or 2 if you're interested. It's a prediction how AlphaFold will change structural biology and molecular biology. And now 1 year later, it's already many of those things we can observe. So for example, that when you solve cryo-electron microscopy structures of large assemblies, and you only have a few of the pieces of the puzzle at high resolution, and the cryo-EM is at medium resolution, you can use AlphaFold to predict the missing pieces of the puzzle and to stick them in and solve the structure of the whole assembly. This is something that is now already done routinely, only 1.5 years later. So here's the summary. We can use X-ray crystallography, also nuclear magnetic resonance to solve structures of domains. Also, if you work really hard and sometimes it takes 5–10 years, you can solve such large structures of complexes by X-ray crystallography, but the effort was enormous to do that, to get it perfectly homogeneous, no flexibilities, so that you can form a crystal lattice. But now with cryo-electron microscopy, you can solve even larger assemblies, and you can solve assemblies that are intrinsically more flexible, because you can sort the particles and you can choose one conformation and solve that structure, right? And you can even solve several structures from the same sample when you sort your particles. Nevertheless, often you don't reach atomic resolution, and then that's when you need these pieces of the puzzle here to solve the structure of the large complex. And there's two main tools that allow you to do that. One is the AlphaFold, which allows you to predict pieces of the puzzle that you don't have. And the other one is cross-linking and mass spectrometry. So why is that important? It's because if you have a low-resolution map of the large complex and you put the pieces of the puzzle together, sometimes it's not totally unambiguous. When you observe enough detail, then it's unambiguous how you put the proteins with respect to each other, right? The orientation will be unambiguous. But often it's not, and then cross-linking can help you, because once let's say you model two proteins next to each other like that, there will be an interface where there's amino acid residues. And when you see cross-linking between the two, for example, two lysine residues that can be chemically cross-linked, and those two lysines are in proximity, then you gain confidence. If your model is like this, and now the one lysine is down here and one is up here, but you actually observe a cross-link, then you know your model is going to be wrong. It's got to be, like this. So there's these tools that allow us to take, structural biology to this level where we have very large assemblies. Now, for your generation, what is next, right? You could say, oh, now, structural biology is done. Actually, just a couple of months ago, DeepMind has published, together with EMBL, a database where you can download now virtually every protein that is known. And by known, I mean that its sequence is known. And you know how many sequences are coming in from the genome projects. And they basically have predicted all the open reading frames, all the proteins that are encoded in these genomes. So it's an incredible resource. But of course, to understand those proteins, how they interact, what their function is, that will be taking at least one more generation to look into this. But another thing is, of course, to understand structures within cells, right? That is now a huge challenge. And one possibility to go about it is cryo-electron tomography. So what is the difference between cryo-electron microscopy and cryo-electron tomography? Well, the difference is that, when you do single particle cryo-EM, you have your sample, you shoot with an electron beam, right? And you record here the image that you get. But you rely on the different orientations of your proteins, right, to reconstruct later the volume. Now with tomography, what you do is you shoot with the electron beam, and then you tilt your sample, and you shoot again, you tilt your sample, you shoot again, and so forth. So you actually get a whole series of images which show the sample from different sides. And that allows you then to reconstruct also volumes. Those will be at much lower resolution, because the trick for single particle cryo-EM is that you average over thousands, maybe sometimes hundreds of thousands, maybe sometimes millions of particle images. But when you do tomography, you cannot do that. You have very little information, so the resolution will be lower. But the advantage is that you then really get a three-dimensional image of your cell, or your organelle that you're looking at, for example, a mitochondrium. And sometimes you can get beautiful images like that, where you see here a DNA, piece of the DNA. You also see polymerases that are shown here in orange, individual polymerase enzymes that are trailing along the DNA. And they are making RNA, which is shown here in red. And at the end of the RNAs, you actually see these little knobs here. These are partial ribosomes that are forming. because what that is actually ribosomal RNA transcription. So with cryo-electron tomography, it was possible to resolve such a process. And this is the new excitement, that we can use cryo-electron tomography to actually visualize cellular processes at the molecular level. And so the promise of this approach here, integrated structural biology, is that we can go, from the atomic details to cell biology, right? To visualizing large machineries of the cell. But we have to integrate the information across these different scales of dimension. And then there's one other aspect, and that is, if you bring in the time dimension, if we can even get snapshots at different times, we can make, and that's the promise for the future, it's for your generation to explore, we can get movies of cellular processes at atomic detail, right? This is what has just begun to happen. There's only very, very few examples. Of course, it's limited to special cases like movement of the ribosome and things like that. But it's becoming possible. This is how far we are at this time. Maybe before I summarize the first part, I want to integrate the information, because that was a lot of information for you, and give you a very simple example. So let's say you've purified a protein complex, right? And it looks like this somehow. And it has, different subunits here that are somehow attached to it. So, and now you have a cryo-electron microscopic reconstruction. And let's say this reconstruction would be at 6 angstrom, right? So this would be your EM reconstruction at 6 angstrom. So here you begin to see some sausages for alpha helices, but the beta sheets are not well resolved and so forth. So what you need is, for example, this structure here at high resolution, right? And so maybe that is, some beta sheet with some alpha helix on top and another one here. And you have that high resolution structure, let's say, from X-ray crystallography. And that probably was at, 2 angstrom resolution. So you have all the atomic details in here. And now you move that into this density, and you've explained it, right? So this is what integrated structural biology is about. And then, because this is the core complex, it's probably less flexible. So these, regions on the outside are probably at 6 angstrom. But this core region may be much higher. It can be even 3 angstrom. This is a concept that is called local resolution. Right? That, a core complex is quite rigid, but then something on the outside can be very floppy. So then you can probably build, that complex here at very high resolution based on the cryo-EM map. And then you need to ask yourself, did I do everything correctly? And you actually would hope to find some crosslinks here in this interface. So that's coming from cross-linking coupled to mass spectrometry. Why mass spectrometry? Because you chemically cross-link to lysine residues, but then you need to map which lysines did I actually cross-link. This you can do with mass spectrometry. So then you know that a certain lysine residue here and a certain lysine residue here are cross-linked. And that gives you confidence that your modeling is fine. So you see how X-ray crystallography, cross-linking MS, cryo-EM work together to finally resolve such large complexes. Is there any questions until here? I couldn't go into the details of structural biology, but it's very important for me that where we are, what is possible nowadays, and what the future will be like. So do you have any questions? No? Okay, then I think we can summarize that part. So X-ray crystallography provides atomic details. It requires crystallization, and it really has shaped, what molecular biology is all about. Then single particle cryo-EM has been there for decades, but over the last decade has made these huge advances. And now is a second method to allow you to get atomic details of large complexes. I briefly talked about tomography, where you actually tilt the sample and you reconstruct volumes, even of organelles or cells, membranes, and so forth. And the machine learning-based structure prediction is now at a stage where it can reliably provide structural information. Now, there's of course this discussion, are these models supposed to be called structures or not, right? So in the end, all the structures we have are models, right? Because we build also models into X-ray maps. It's just a question, how well supported are the models by experimental data? And in case of AlphaFold, they use the whole protein database to train the algorithm. So obviously, a lot of experimental information has been used to learn some of the rules for how proteins like to fold, and then it's applied and gives you a model. So, this whole discussion is actually not so meaningful. The way I do it, when it's experimentally, an experimentally defined structure, I call it a structure, but the actual model that you deposit remains a model, right? But when it's coming directly from an experiment, then I call it a structure, and otherwise I talk about models. And that many people do it like this. But in the end, you have to be aware of the reliability of your molecular model. And for that, you always need to ask, where does it come from? Which assumptions were made to get this model? How was it refined? What kind of experimental data did I have? And so forth. And then, of course, NMR that is also possible in solid phase. And it's normally limited in size, but has this advantage that you can look at binding events, you can look at folding events, you can look at flexibility. And there's other methods like cross-linking mass spec I talked about, but also small angle X-ray scattering that can give you shapes of structures, or fluorescence resonance energy transfer, which uses fluorescence to measure distances between molecules, and so forth and so forth. And actually, light microscopy is on a good track. We have here the group of Stefan Hell, and they are getting better and better with resolution, and they really reach now molecular resolution also with light microscopy. So this gap that you currently have between cryo-EM, X-ray, and light microscopy, this is beginning to close. And so we will soon have a kind of a continuous imaging of cellular processes. So it's a fascinating time to be in the field. Maybe finally, before we have a short break, what did structural biology do during the corona times? I started with corona and the mRNA vaccine. Now just briefly, what did structural biology achieve? It was so interesting to witness in real time how fast the world would move, the world of science would move. And within, a year or so, we had all the structural information about all the coronavirus proteins. And our laboratory also contributed to this. This is actually the structure we solved in the lab. It was an interesting race. There were 3 Chinese teams and our team. And within a week or 2, all the 4 groups published the coronavirus polymerase structure, the first visualization of how the virus actually replicates its RNA genome. And why is that important? It's because, some of the drugs, actually the first drug that was approved, remdesivir, but also molnupiravir, they are interfering with replication by the corona polymerase, and we could visualize that. And this is briefly what I want to show you. So this is the structure of the polymerase, and now we can actually see how remdesivir interferes with the process of replication. So it's a nucleoside analogon that is incorporated into the growing RNA chain, but the polymerase doesn't stop. It actually goes on incorporating 3 more nucleotides. One is here, the next one, and a third one. And only then the polymerase is pausing, and this is because there's 2 atoms here in the remdesivir moiety that clash with the serine side chain. And you know, that has an advantage because remdesivir escapes the so-called proofreading activity of the polymerase. It's a drug that is not made useless by the proofreading activity, because most antivirals would stop the polymerase immediately, and then the so-called proofreading activity would cleave off the last nucleotide, and the drug is basically useless. But remdesivir, because it stops it with a delay, has some efficiency and is working as a drug to some extent. But it's not a perfect inhibitor, because due to thermal motion and if there's enough nucleoside triphosphate substrates, this barrier can be overcome and the polymerase will keep going. And also you have to give that drug very, very early after infection, when the virus is replicating. After that, the patients have other problems. It's too late. So there's issues that need to be solved. And if you look at molnupiravir, the other drug, you see it's interfering with the same process, but its mechanism is just entirely different. So it has been incorporated, this molnupiravir has been incorporated into RNA, and now that RNA containing the drug derivative is used as a template. And when that is used as a template in a second step of RNA synthesis, it can lead to errors, because it can form 2 types of base pairs. You see here it's base pairing with a G, but it can also base pair with an A, and that actually introduces errors during replication. So the way molnupiravir works is it's not stopping the polymerase, but it's introducing so many errors during RNA replication that the virus is not viable. Yeah, this was the very first part, and what I would suggest is that unless there's questions now concerning the structural biology, that we do a 5-minute break, maximum 10 minutes, and then we will go straight into discussing transcription, polymerase and transcription initiation. Thank you very much. Okay.